Good evening, everyone. It's a pleasure to be here. Thank you to the organizers who put this on. I'm really excited to share with you all my work on immune-acting fibroblasts and their role in skin inflammation and host defense. And I am a PhD candidate in Rich Gallo's lab at UC San Diego. So let's talk about skin. Skin infection by Staph aureus. We've already heard about these tissue and the bacteria a little bit today, which is good. We got a primer already. Staph aureus is a gram-positive bacteria responsible for most skin and soft tissue infections in humans. I'm sure we've all heard of MRSA, methicillin-resistant Staph aureus. This antibiotic-resistant strain has really complicated treatment, and this has led to a lot of morbidity and mortality. So it's really important that we understand the host defense response to this microbe so that we can develop effective therapeutics. So what do we know about the host defense response in the skin against Staph aureus? The thing I'd like to focus on today is the cytokine IL-17 that is produced by subsets of lymphocytes. This cytokine is thought to work in two important ways. First, it is thought to work on, one behind here. It's thought to work on epidermal keratinocytes and drive their production of neutrophil chemokines, antimicrobials that kill the Staph aureus. Second, it is thought to act on these epidermal keratinocytes and drive their production of neutrophil chemokines that bring neutrophils into the tissue to kill the Staph aureus. And we know that neutrophils are really important because when you don't have them, when you're neutropenic, you probably die. So we need those. In 2015, our lab made a really novel discovery in terms of defense against Staph aureus. We identified a new cellular player. We found that during invasive Staph aureus infection, subsets of fibroblasts in the dermis, in particular in the deep reticular dermis, would differentiate into fat cells, and we get this huge expansion of fat in the dermis. And we called this process reactive adipogenesis. And importantly, during reactive adipogenesis, the immature adipocytes would make cathelicidin, this antimicrobial peptide that is very important for killing Staph aureus. Now, in light of our study and other cell atlases studies more recently, we've changed how we think about fibroblasts. We used to think of these cells as simply structural cells in the skin. On the one hand, you have the classic fibroblasts making matrix and promoting scar formation, and on the other hand, you have pre-adipocytes that turn into fat. But we now recognize that these cells are incredibly diverse with many different cell states and types. And in addition to the functions I've mentioned already, it's been observed that many of these cell states and types have possible innate and adaptive immune function in the skin. But there's an important gap here. We don't know if dermal fibroblasts as a whole or any subset play a significant role in skin inflammation and host defense. So this is what I set out to address with my thesis project. And to address this, I infected mice intradermally with Staph aureus, a MRSA strain, and did single-cell RNA sequencing. And I used these data to perform an unbiased machine learning network analysis to understand cell-cell communication during infection. And what I found was really surprising. Fibroblast to myeloid communication was the dominant pathway observed during Staph aureus infection. And I say this is surprising because based on those models of pathogenesis we saw previously, you would think keratinocyte to myeloid communication would be the strongest. But no, it was the fibroblast. So to understand how fibroblasts might be communicating with myeloid cells and more specifically neutrophils, we looked at the differentially expressed genes in the fibroblasts in our data set. And we found that these cells during infection upregulated many different neutrophil chemokines like we've heard about today, CXCL1, 2, 12, many different pro-inflammatory cytokines and growth factors. And overall, this transcriptomic change was consistent with IL-17 signaling. And remember, this is that key central mediator for defense against Staph aureus. And importantly, the contribution of fibroblasts compared to other cell types in the skin in terms of expression of these chemokines was very high. Now, I mentioned fibroblasts are becoming appreciated as heterogeneous. So we looked at our fibroblast subsets in our data to understand, which ones are communicating with neutrophils and myeloid cells. And at this given resolution, we clustered 11 different subsets of fibroblasts. And I've drawn a line down the middle of this UMAP I'd like you to pay attention to, defining groups of clusters, 1 through 7 on the right and 8 through 11 on the left. When we look at the neutrophil chemokine expression, we can see that expression of these genes is largely restricted to clusters 1 through 7. And the expression, importantly, is heterogeneous within this group of clusters. We see CXCL12, which is broadly expressed by these fibroblasts, whereas other chemokines like CXCL5 are restricted to specific clusters. Now, there's many different well-documented fibroblast marker genes, those that mark adipocyte lineage fibroblasts and those that mark those more classic matrix-producing fibroblasts. If we look at the expression of these genes, we can see that the chemokine-producing clusters largely express adipocyte lineage markers, but not those classic ECM markers. So we've defined the fibroblasts of interest to us as CXCL12 positive adipocyte lineage fibroblasts. And to confirm this through more unbiased method, we look at the GO term analysis between these two groups of clusters, and we see again genes related to fat cell differentiation in that CXCL12 positive cluster and not in the other cluster. Additionally, we see this CXCL12 positive cluster talking to neutrophils more than the CXCL12 negative clusters. And importantly, the CXCL12 positive one is talking to neutrophils more so than non-neutrophil myeloid cells. So really specific for neutrophil communication. Now, to understand where these cells exist in space and to confirm our single cell data, we perform confocal microscopy. And here we're looking at PDGF receptor alpha, which is a pan-fibroblast marker, and the neutrophil chemokines CXCL1 and CXCL12. We can see that during staph infection, CXCL1 and 12 are upregulated, but not in all fibroblasts. It's really specific to the fibroblasts that are in the deep reticular dermis, which aligns with our single cell data. So with these observational data, we've developed a hypothesis. The CXCL12 positive fibroblasts that are adipocyte lineage in the deep tissue are activated by IL-17 to recruit neutrophils for host defense. Now, to address this hypothesis, the first question we ask is, can we model this in vitro, this communication between the fibroblasts and the neutrophils? So first, we took 3T3L1 fibroblasts, and we chose this cell line because it is a pre-adipocyte and will differentiate into fat cells in vitro. And we stimulated it with IL-17 among other stimuli involved in host defense against Staph aureus. And then we looked at neutrophil chemokine expression, and what we found was that IL-17 on its own didn't do too much, but when combined with other signals involved in host defense, it potentiated these signals. And importantly, we saw a huge induction of neutrophil chemokine expression by the combination of IL-17 and TNF. And so we chose to focus our later studies modeling the fibroblast activation. We focused on this combination IL-17 and TNF. And to understand more broadly how these cytokines activate pre-adipocyte fibroblasts, we performed bulk RNA sequencing, and we also compared the effect of these pro-inflammatory cytokines to the effect of adipogenesis, which we had previously shown induced antimicrobial expression in fibroblasts. And what we found was a lot of different host defense genes that are of interest going up with the combination treatment of IL-17 and TNF, but unique to IL-17 and TNF and not induced by adipogenesis. What I'd like to talk about today, because of this, the importance of migration with neutrophils, are all the chemokines that we see induced. So, the next question for us is, do these cells in fact recruit neutrophils? So we performed a trans-well migration assay, which you guys heard about previously today, where we took our pre-adipocytes, stimulated them with IL-17 and TNF, took the conditioned media from those cells, put it in the bottom of a trans-well, and then put mouse bone marrow cells on top, which include neutrophils among other leukocytes. And then we let the cells migrate across and enumerated the different cell types by flow cytometry. And what we found was that when fibroblasts were not stimulated, they induced some migration of neutrophils, monocytes, and lymphocytes. But when we hit these things with IL-17 and TNF, the fibroblasts were very potent at inducing neutrophil migration. They also induced monocyte and lymphocyte migration, but the important thing to look at here is the fold change, which you see is far greater in the neutrophils compared to other cell types. Now, what is the mechanism? There's been many purported neutrophil chemokines. Here I'm showing you that inhibition of simply the two receptors on neutrophils, CXCR2 and CXCR4, we can completely eliminate the neutrophil migration towards the pre-adipocyte fibroblasts. So not only is CXCL12 a good marker of our fibroblast subset of interest, it is also a functional marker. So the big question, and if there's one question you're going to pay attention to for this talk, it's this one, like what's the importance of all this? Does it matter? We address this by first developing mice that lack the IL-17 receptor specifically in fibroblasts, and we did this by crossing PDGF receptor alpha Crea mice to IL-17 RI flox mice. And then we put these mice through our intradermal Staph aureus infection model, and here we used a reporter strain of Staph aureus so that we could non-invasively measure the Staph aureus load over time. And in separate experiments, we harvested the skin at day 2 to look at other endpoints. And what we found was that in normal mice, right, that have their IL-17 receptor intact in the fibroblasts, we see increases in neutrophil chemokine expression and antimicrobials as expected. But when we use our transgenic animals that lack the IL-17 receptor specifically in fibroblasts, we see significant reduction in neutrophil chemokine expression. In line with this, we see a striking reduction of neutrophil recruitment to the dermis in the transgenic animals. And this is the non-invasive imaging here. We can see that with the orange line here, the transgenic animals have greater bacterial load, which is consistent with having less neutrophils in the skin. And all of this leads to larger lesions in these mice. So we've answered our question, right? Fibroblasts are critical for neutrophil inflammation and host defense, and their recognition of IL-17 is a major determinant of this process. You know, as is typical in science, the answer to one question leads to many more, and the most obvious next one to us is, are these cells also important in non-infectious IL-17-driven inflammation? So let's switch gears now and think about psoriasis. So psoriasis is a chronic inflammatory skin disease, but it's also characterized by neutrophil inflammation. It is also thought to be driven by IL-17 and its effect on keratinocytes and their production of antimicrobials and neutrophil chemokines. And thankfully, we have a good treatment for this disease. The IL-17 inhibitors are very effective, and they are thought to work by inhibiting keratinocyte activation. And importantly, the role of fibroblasts in psoriasis, like in Staph aureus infection, is unknown. So we used our mice that lack the IL-17 receptor specifically in fibroblasts and challenged them with imiquimod topically. This is a very common psoriasis model. And what we found was very similar to the Staph aureus infection, significantly reduced neutrophil chemokines in the transgenic animals that lack IL-17 receptor in their fibroblasts. And in line with this, we see significantly reduced neutrophils in the skin of these mice. So fibroblasts and their response to IL-17 is not only critical for Staph aureus infection defense, but also for neutrophil recruitment in non-infectious inflammation. So do these cells play a role in human skin disease? To address this, we recruited patients that are on and off IL-17 inhibitors. Like I mentioned, these are very effective at treating psoriasis. And we took lesional biopsies from these patients and stained them for the neutrophil chemokines CXCL1 and CXCL12. And what you can see in the untreated patient, in the dermis, what I'm highlighting is fibroblastic cells highly expressing CXCL1 and CXCL12. But when these patients begin taking IL-17 inhibitors, these fibroblasts are turned off. They're no longer making CXCL1 and CXCL12. So this suggested to us that IL-17 inhibitors may work by inhibiting CXCL12 positive fibroblast communication with neutrophils. So in summary, what I've shown you today is that in the skin and inflammatory contexts like Staph aureus infection and psoriasis, pro-inflammatory cytokines like IL-17 and TNF synergistically activate dermal fibroblasts, and specifically the CXCL12 positive adipocyte lineage subsets that exist in the deep dermis, driving their production of CXCR2 and CXCR4 ligands that recruit neutrophils to promote inflammation and host defense. So some conclusions and final thoughts. If there's one thing I'd like you to remember today, it's that fibroblasts are diverse and multifunctional, and we are now thinking about this subset that we've defined as immune-acting fibroblasts, or IAFs. And these cells in the deep dermis protect against Staph aureus infection and contribute to psoriasis pathogenesis. And they do this by serving as receivers and transmitters, really integrating a lot of local signals, and possibly serving as the last line of defense deeper in the tissue. And so we've made a lot of important advances with this work, but there's still a lot to do. And some of these questions that we still have, for example, are what are the role of these cells in other inflammatory contexts and other tissues? And how can we therapeutically target these cells specifically? So with that, I'd like to say thank you to all of those who contributed to the work, the funding sources, and thank you all for listening. I'm happy to take any questions, and while I do, I'm going to advertise my side project, which is my podcast, so thank you.
So in your psoriasis model, you only show the neutrophil recruitment, but not the disease phenotype. So did you really see, based on what you said later, it seems that it will also impact the disease phenotype. So if that's the case, what is the role of keratinocytes that people that thought should be the target rather than the fibroblast?
Right. I would argue that the neutrophils are part of the disease phenotype, but we're really focused on the mechanisms of inflammation and host defense. So we really focused our analysis on that endpoint. I have an impression that it is not significantly different, but I need to quantify that to be certain. But it's not our focus.
Okay.
Thank you.
One quick question, Kellen. I have a similar question as I did to Gregory. Do you think that this axis, these fibroblasts, are also regulating or talking to neutrophil functions in the tissue, like persistence and other functional?
Yeah, I didn't have time to go into some of that today, but we also did some in vitro assays looking at neutrophil function, and we see that fibroblasts are inducing classic neutrophil activation phenotypes.
Beautiful work, really.
Thank you.
So my question is, what happened to those fibroblasts in IL-17 signaling? What is the fate of these cells in the context of inflammation? You show the absence of chemokine production, but they may be transformed in something really important for the protection.
Yes. So this is not something we've done, but something we are working on doing. So I plan to do fate mapping using a CXCL12 Cree mouse and just test whether do those cells in fact turn into fat cells. They express all these adipocyte lineage genes and are deep in the tissue where you would find fat cells, but it's unclear if they actually will turn into fat or not.
It's really fascinating to think also in the context of cancer, actually, what 17 signaling on fibroblasts could actually just do there, and actually how the absence of this. Anyway, very interesting.
Absolutely, thank you.